So, hello everyone. I think everything seems to be working again. Let me quickly check. Yes, I think I'm online. Okay, yeah, hi, and welcome to another Saturday live stream. Yeah, happy that you could join me again. I've seen that several of you already started commenting here. It's always quite nice to see people from all around the world joining in here. Well, I'm doing the live stream, as you already know, from Central Europe, from Austria. It's snowing right now. It's pretty cold. And I've read in the news that, of course, in other parts of the world, in Australia, for example, it's quite the opposite. It's pretty hot. So still, we're all happily united here together, doing microscopy together. Today's topic, I have to tell you a little bit, is not an easy topic. I've been thinking about doing this for some time now. I know it's, but as always, if you have any other suggestions, that you want me to put things under the microscope, I can. I do have a couple of live samples here as well, which I also want to show you. But I do want to talk a little bit about the fibers as well, dust fibers and the microplastic fibers and so on, because they do appear quite frequently. And I did get a couple of, as always, emails and questions and comments where people find those fibers and they kind of want to know what it is, right? And in many cases, these are, of course, natural fibers from plant material or so, from clothing a lot, and sometimes also synthetic fibers. And I thought we're just going to have a look at a couple of samples that I've prepared today. And we're going to basically take it from there. And then please feel free to write comments into the chat. And then, we can, I can try to pick up on those. Yeah, I think I'm just going to make the hello short. As always, from Austria, from Oregon, from Australia, from Northern Germany, Florida, United States, London, England, New Jersey, Upper Austria. Okay, very nice. Okay, so what you see over here is already a live image. You can see that there are some bubbles in here, and these are fibers from my clothing. So basically, where did I collect them from? Last week I already told you that I collected a few of them. Yeah, they collect inside the belly button over a couple of hours. Basically they come off from the clothing, and this already shows something quite interesting, and that is that if there's synthetic fibers in clothing, nylon, polyester, and so on, then this actually is indeed contributes quite a bit to microplastic pollution as well. Natural fibers, of course, are not a big problem like cellulose fibers, cotton, for example. But there are, of course, also synthetic fibers around that essentially can cause problems, especially when they're taken up by organisms. Yeah. So this is a little bit. So what I've got here is I do have some synthetic fibers as well that we can look at, and I've got some dust samples, and I just thought we're gonna kind of take it from there. Okay. And I've also got something already set up. And this is a soldering iron, okay, 300 degrees, and with a very sharp pointed tip, and with a soldering iron, you're actually able to test whether a microscopic fiber is actually synthetic or natural, because the synthetic fibers they're able to melt, and hopefully we're able to also be able to see that. Okay, so that's kind of a little bit the idea of today, and we're just going to take it from there a little bit. What I want to do, however, as well, is I want to show you also some kind of different imaging techniques. So this one is now DIC. In bright field, it looks, of course, a little bit less exciting, maybe less colorful. However, bright field does have the advantage that you're able to see the natural colors of the fibers much better, because some of them are actually stained and colored, and this can, of course, be seen much better when you're looking at it without any filters or so. And of course, if you have a stereo microscope, then you can also use a stereo microscope in cross-polarized filters, and I also want to show you that. So let's go over here, and we now need a light from the bottom. Okay, so let me turn that on. We've got now light from the top. We need to switch this off, and we can turn on the light from the bottom. Okay, and I'm just going to do a little bit of cross polarization, like you've already seen before. I'm just going to drop now the fibers here on top, This one goes across like this here. I have to refocus again, put the microphone a little bit closer. Yeah, and basically, yeah, it's a little bit the cheap way what I'm doing right now, because I simply dropped the filter directly on, yeah, on the slide. Yeah, this is how it looks like. It's a little bit overexposed now, so let's zoom in a little bit so that the camera is able to, yeah, and then I'm going to refocus again, and this is how it looks like. So that now you also know how I got the thumbnail image, okay? So that is actually one of the nice things here, and I'll be also using the stereo microscope then to test a couple of fibers of whether they're natural or synthetic, okay? Yeah, so, greetings from Vietnam, from sunny Southern California, yes, yeah. That's a little bit one of these things is that because of course I've got a camera connected, it always readjusts the exposure. So you see that yeah, if it's a little bit like yeah, a little bit right now some parts are a little bit overexposed. Yeah, so the image quality as you look through it, of course, isn't much nicer. See now it kind of sticks. The slide sticks to this here. Okay, so just yeah, as a little short intro, I just wanted to show that to you. Let me quickly put this back here. Okay, wipe it off. Let's switch over again to the other microscope, and here we are again. Okay, yeah, we can also try to have a look here. When you're looking at fibers, what I sometimes see is the following: that yeah, there can also be hair in there, of course. And what I would, however, yeah, do next is I'm just going to show you a couple of other samples that I've got. And the other one that I want to show you is the following, a water sample, which is also a little bit interesting in the sense that it kind of shows how much pollution this causes. And this is from a washing machine. So I've collected a little sample of washing machine water. And then I do also have over here, yeah, also something that I would like to put under the microscope. This is artificial meat, so vegan meat, and unfortunately I was also able to find a couple of fibers in there. I say unfortunately because I think they should not be in there, but this has been actually found also by other researchers that unfortunately a lot of yeah, I would say highly processed foods, also vegan meat and so on, unfortunately also contain a lot of these fibers, microplastics as well. So this one over here is now some water from my washing machine. Okay. Yeah, there's a very common, quick comment here. How can you find out if with some fibers are natural, synthetic? I'm going to show that to you with the with the soldering iron. Okay. So, but I just want to show you yeah, a sample from over here. And this is actually also something that some researchers have been investigating a little bit. This is the yeah, the fibers that come off from clothing, and this is actually over the washing machine. The problem with that is that those synthetic fibers from clothing, obviously they are not broken down when the water is processed, and therefore they end up in the environment. And that's, of course, a problem. So this one is now, let's see if we're able to find a few things here. This is basically just some water from a, yeah, from a washing machine. Okay, so, and it could be that some of them actually, let me quickly check. Oh, that's an interesting one. Because, where are they? Just a second. That's what you call demonstration effect. Because, why do I not see anything? I used to see so many of them. What I think could have happened is, here, it's only one. Yeah. What I think what has happened is sometimes what they do is the fibers have a different density than the surrounding water, and so they either float up or they sink. Okay. I hope I took the correct one. Not that I have basically shown you the. Let me try this here. Okay. Okay. Oh my gosh. Okay. So this is what you call a demonstration effect, because this is actually I see so much stuff floating around here, but I was not able to catch anything here. Okay, that's kind of interesting. Okay, let me try this again. You see, I tried this before. I found so many fibers, and for whatever reason, not anymore. That's what you call a demonstration effect. Okay, yeah, but here we go a little bit here. It's a little bit better. A lot of gooey stuff, and this is actually the thing that I also wanted to show you. That some of the fibers, what you see, they seem to aggregate and accumulate other things as well. Yeah, so you see that there are other particles sticking around or sticking on the fiber. Yeah. And what you normally can do is the following. You can try to separate them by doing a so-called a flotation. You add salt to the water, and then many of the fibers are less dense, and they will start to actually go up and float on the surface. Yeah. So you see essentially, yeah, all of this stuff that was not only dirt, but of course also from the clothing, yeah, things that have rubbed off. Yeah, and started to then accumulate. Yeah, I mean, honestly, I have to tell you, this is not such a nice sample because I've had already collected it from my washing machine. I found so many; it was completely full. Yeah, so I would say it's probably a sampling thing. Yeah. There are, as always, a couple of, how do you say, off-topic questions, like, for example, what camera would you recommend? What I will do is the following, is I want to answer those questions, which are a little bit off-topic, towards the end of the live stream, like I've been doing always, okay? So this is just a little thing that I'll put over there. So what I want to do now is I want to show you some cotton, some regular cotton that I've got here. Okay, with a polar, you know what? That's a good point. Let's try that. I'm going to now do the following. I'm going to thank you for the comment. I'm going to now do the following. I put it now again over here, and I'm going to now hold the I can thing over it. You see that I have a problem now. How am I gonna hold this over that here? And I have to somehow refocus this. Let me do the following. Let me zoom out. Okay, here we go a little bit. Okay, and it's a little bit out of focus. Yes. Yeah, so basically, yes, it works better, right? Because you're seeing it with the contrast much better. So I put it now in the cross-polarization, right? Let's not forget that this is just a small sample, obviously, yeah? And those fibers here, if they're synthetic, of course, they constitute a significant part of the so-called of the microplastics problem that we're seeing nowadays. Yeah? So those fibers essentially are so common these days, also in dust and so on, also being a natural part of the environment, that essentially they can be really annoying when you're doing microscopy and then you see those fibers. People actually already posted online and sent me emails. They, for example, they observed their own blood sample under the microscope and they saw those fibers and they were kind of worried that this is kind of in their blood, but it's not, right? It's just basically a contaminant on the microscope slide. Yeah, so that's basically. So let's do the following. Let's put a few things here as well. Let's start off with this one over here that says just some regular cotton. Okay, as always, I put a little bit of cotton here. I need to. I also always want to use a clean tip. By the way, I don't know if you've been realizing this, but I've had some technical issues, not only with my forum, but also with my webpage. It's kind of weird if I say it. My website is actually under attack. Some bots, automated bots, are trying to take down my website. So I've got hundreds, if not thousands, attempts, automatic attempts to access my website, because in the logs I was able to see that. And this sometimes gives an error message when you try to visit the website or the forum. I just want to reassure you that I'm aware of this problem, but yeah. Hundreds of different automated bots are trying to kind of get access to my website, and this takes down the server. And that's a little bit of a problem that I have had again recently. And so sometimes you get an error message when you visit the forum or the web page that it's not able to reach. Then you simply have to reload it and hope that it's going to work then. So this is that's cotton. I need to get the scope view. And the cotton over here, look at it looks extremely nice and regular. Okay. And so this, I just want to show this to you because actually cotton is cellulose. Yeah, it is actually a natural fiber. Yeah, and it looks kind of pretty. But it also shows in a that even though it's cellulose and a natural fiber, it looks extremely regular. And the reason is that the cotton is made synthetically. It's still a natural fiber, but it's artificially made. So there is actually what we've done a long time ago. I remember when I was still at university, we used to make our own cotton. We did a chemical reaction, and then the molecules, the glucose molecules, start to automatically polymerize, and we actually made cotton in a test tube. So this is kind of a polymerization reaction, but as we can see, it looks quite different. Let's add a little color, from the other fibers that we see. So that does my DNS hosting provider offer an anti-DDoS service. I did not contact them yet. I know this is a little bit off topic, but it was like this. When I had the first problem, I contacted them. They immediately responded, and they basically told me what the problem is, and they blocked a couple of the IP addresses. And now the problem started to reappear. And every time when I block IP addresses, new ones keep on appearing. So it's really hundreds. Yeah. So this is actually quite nice looking indeed. Okay. So let's do the following. Let's have a look again under regular polarized light. I need to, yeah. See, again, this is a problem of overexposure, right? So it's kind of a little bit blown out, and it's why I have to go down a little bit with the light intensity. So this is what I call modern art. So let's have a look again at the stereo. So I've got my simply polarization filter on the bottom here. And then now I'm just going to hold the other one on top. Okay. So here we go. And we can zoom in a little bit. That's regular cotton. Okay. So I would like to simply try now the following. I'm going to just use now normal cotton. And let's try the soldering iron test. Okay, which I have to tell you, I did not try it on cotton. Yeah. I'm just reading the comments here as well. So I'm gonna contact them in any case. I mean, the situation already improved a little bit, but just in case you're getting this weird error. Then you have to click the reload button, and then the website will work again, maybe after a couple of minutes. But it was really annoying. I spent quite a few hours on this, trying to configure my web server in such a way. So the desk view. So let's try it. Okay, it's got a little bit, and So let's try it. How am I gonna try this? What's the best way to try this? Well, I think I'm just going to put a little bit of this cotton here. I'm thinking what the best way would be. You see, if I put cotton directly on a slide, then there is the problem that it's not flat. So what I think I'm going to do is the following, is I'm going to dissolve the cotton, or suspend rather the cotton in a little bit of water to make sure that it's kind of flat, and then I will dry it again on my coffee warmer. Let's see if this is actually a better way, because I want to make sure that the cotton fibers, that they're completely flat. Okay. So I take this here. I add a little bit of water. Again, I take a little bit of cotton as before, not too much. And you see, when you put it in water, then it kind of sticks to this. Then it sticks to the slide, right? And that's actually what I want. Yeah, and then I'm just drying it right now. Yeah. So the water thing is only that it's kind of flat, okay? Because I need it to be accessible with the soldering iron. Okay, and you see it's already drying. Maybe we can make it a little bit hotter, 65 degrees Celsius. Yeah. And this soldering iron technique is not something that I invented or so. I did a little bit of research, and some publications that I found actually said that this is an official way of, official under quotation marks, way of testing the material of the fibers. And you have to use a hot needle. So basically a pin or a needle. But I was kind of saying, well, I mean, if I put a needle over a candle and by the time I've got it under the microscope, it's kind of already cooled off. So I just am trying to use it with a soldering iron. Okay, so it's now, you see, it's now completely dry, right? But the cotton is flat on the slide. What we can see, however, are those water spots, right? The calcium carbonate, which we're just going to ignore. Okay, I'm just going to turn this off again. See, it's still kind of hot, so I'm going to put it somewhere on the table to cool it a little bit. Okay. Yep. So, and I'm going to now this is where the contrast is probably going to be a bit bad. Okay, so this is now, let's see. Again, I have to focus this a little bit. It does kind of work. Okay, so that's kind of the cotton right now, right? So, and the soldering iron has been set to what, 300 degrees. Okay, I better not burn myself. You can maybe follow a little bit at the top what's going on. I know it's a little bit, again, a little too dark, but you see I've got the soldering iron here. And I will try to do the following. I will try to now touch the cotton. Okay. And what they actually say is that if you touch it, you see that it doesn't melt, number one. And number two, if you keep on doing this, it will actually start to char and turn brown, obviously, because it's going to burn, right? Yeah. But I think it's kind of very clear simply from the behavior. Yeah, and you can actually maybe already see it slowly starting to change color, starting to become fried a little bit. Yeah. Yeah. So this is a sign that we're not talking about, how do you say, some kind of a plastic. Yeah. There are also certain stains and dyes that you can use because plastic synthetic ones are hydrophobic, right? And therefore certain stains will actually adhere better. Yeah. Yeah. For those of you just joined in. What I'm doing right now is this is under my stereo microscope. This is cotton, and when you basically, I'm touching it now with a hot soldering iron, and therefore I always have to go off a little bit because I don't want the heat to go all the way to my microscope lamp. Yeah, but you can see already that it starts to change color, obviously, right? Yeah. Let me see what happens if I actually turn on the light from the top. Not a lot of difference. Oh yeah, maybe. So that's now top illumination, light from the top. You know what I'm gonna try? I'm going to try to make the how do you say, the bottom side a little bit darker. Where did I put my polarizing filters? Can you believe this? I just had them and I just put them somewhere and I forgot where I put them. This is really cool. I mean, I just had them. Where did I put them? On my table. I keep on losing things directly on my table. Isn't this cool? Wow. I just put them somewhere and I totally forgot. You see the two polarizing filters that I had just a minute ago? I lost them actually on my table. This is something that really didn't happen before. It's kind of funny. Maybe it'll reappear. This is really strange. I don't know. You see, sometimes when you're a little bit with your mind somewhere, you put the things somewhere and then all of a sudden, it's gone. Here they are, right next to my microphone. I put them directly on the microphone stand. Yeah, black on black, and I could not see it. So let's do the following. Yeah, see, that is, I put a polarizing filter on the bottom, simply to make it a little bit darker. Yeah, and then you can now see already that it's a little bit more, how do you say, it turned a little bit brownish. Yeah? Yeah. Because I'm kind of frying the cotton here. Hiding in plain sight, that's exactly. Yeah. So I think you get the idea, right? And I've tried doing this also with real individual fibers that I found. This also kind of works, but the difficulty is that the tip itself is not fine enough, even though it's quite pointed. Yeah. So I was kind of thinking maybe I could somehow connect a needle or a pin or something like that to the tip of the soldering iron. But I wouldn't know how to do that. Yeah. Maybe with a little bit of wire or something. Yeah. So, but let's try now the following. Let's try now a different one. So these were basically cellulose fibers. And now the next thing is the following. I don't know if you know what this is here, this cord. Oh, why do I show it to you here? I can show it to you here. This is called Paracord. So some people actually use it to make arm bands and things like this. And if you just look over here, then yeah, it looks like this. And these things here, they look very, very, very synthetic, which is actually a nice specimen then. So I'm going to take a little bit of this stuff here. I'm going to put it directly on here. Okay. We can actually, yeah. Later on, maybe we can try to put a cover glass on it as well. And let's have a look. It's not flat now, but let's see how this works. Okay, I mean, it already looks a little bit different. You know what? Why don't we try the, how do you say again, the light from the bottom? Let's light from the bottom, and let's put again, cross-polarizers on top. Look at this. I love this. Look at the colors. Yeah. Yeah, this is basically what plastic and so on it likes doing. Yeah, look at the beautiful colors here. This is already a sign that we're not talking about cellulose here. Okay. So these are again these are white fibers. Okay. Yeah. And so this can indeed also be used a little bit to differentiate the natural one, and they look way more regular than the ones that I showed you at the beginning, right? Huh? So let's do the following. I mean, this is kind of obvious what I'm doing right now and what we can expect. I mean, you actually don't even need a microscope for that. Okay. Okay, let's see if what happens when I touch those. Look at this. I think, yeah, the effect was very, very clear. Yeah, it completely melted, right? Yeah. So this is, yeah, if we want, we can try it again. I need to just take some tissue paper and wipe the how do you say, the soldering iron. So, between artificial cotton and cotton. Ooh, I only have the cotton that you use for kind of cleaning your face right now. But I mean, when you look at plant cotton, that would be actually an interesting one. Yeah, maybe we're able to get some. I don't know. Do we have from a cotton plant? That would be interesting, yeah, to see the differences. So I need the string again. See, now the same thing happened with the string. It's on the floor. Okay, let's give it another try here, just for the fun. We can actually try to maybe also take some of the other, how do you say, from the pigmented one. Okay, this is the one here, and let's take a little bit from the colored outside as well. So, and let's try this again. And then later on, I'd like to make a proper mount, and then we're going to see how this then looks at high magnification. So again, under the stereo microscope, let's have a look here closer. It would be interesting to see now if this actually also it also looks kind of nice. Yeah, this is now the colored one. Okay, I mean, you get the idea, right? And here, this one over here, that's the one, the white one. Okay. So let's zoom out again a little bit. Yeah, and this is basically something that I also tried to do. I can show you later what I've done. Yeah, and let's, if you go again here, and if you touch the individual fibers, yeah, it is way too fast. Yeah, it melts, obviously. Yeah. Yep. That was a very quick response. But this is actually nice. Here we've got an individual one here. Where is it? So let's test this. Yeah, it quickly melted. Yeah. And so that's one way how you can go through your fibers and to test them what they actually are. Yeah. Let's do the following. Let's try the following just for the fun of it. Most of the fibers, however, might not be synthetic. They might be natural, but I do have a dust sample here. And let's do the following. Let's try to, so that's basically just some dust from my desk. So I'm just going to do the same thing as before. Add a little bit of water here. And I'm going to just suspend a little bit of this dust, which is probably going to contain a lot of other stuff as well besides fibers. And what I want to do now is I want to really spread this apart, which can be a little bit difficult sometimes because it likes to stick together so much. Okay. And then, yeah, I'll move on and I'll put the other thing here on the microscope as well. So let's, yeah, let's just assume that this is finished. I'm going to put it again on my hot plate to evaporate the water. Here we go. So I'm going to, while it does this, I'm going to read a little bit some of the comments that you've got. Yeah. Asbestos. Yeah, asbestos fibers, honestly, yeah, I've seen sometime and read somewhere, indeed, there are different types of asbestos fibers. However, I'll be honest with you, you probably don't even have to observe this under the microscope. Yeah. You probably just by looking at it and so on, you should be able to find. What I disadvise somehow is trying to cut things apart as asbestos, because the asbestos dust is you shouldn't be inhaling this stuff, as we know by now. But I know that asbestos under the microscope does have a specific looks in a specific way. But I mean, honestly, then you'd have to kind of scratch stuff off from the, how do you say, from the garage roof, or you'd have to cut it, and you probably don't want to do that, yeah? Because it's the asbestos dust which you don't want to inhale. Yeah? So okay, so now it's dry again, Okay, maybe still a little bit too much together, right? But it doesn't matter. I'm going to, yeah. Let's try it here. So this is just dust from my table. You need the stereo view again. Here we go. So this dust from my table. Let's have a look here first. And you see that the fibers are pretty, well, quite a bit smaller actually. So let's do it again on the cross-polarized. Okay, you know what, I'm gonna do it now. I'm just gonna drop it now directly on here. Look at this. Yeah. You see, that's a little bit of problem with the camera, which adjusts the exposure. Yeah. So that means they look a little bit overexposed. But we can already maybe identify that some of them actually do change the color on the polarized light. Yeah. Yeah. Yeah, I'm at the same time reading again a little bit the comments here. Yeah. So, there was, I think, the comment or the question, why are some fibers colored? Indeed, some of them are colored because blue and so on, because they're from clothing. Yeah, the house dust does indeed contain a lot of fibers from clothing and also skin cells. Skin cells are very, very common. So, and you know what I'm going to do now is I'm going to zoom out again a little bit so that we can see this better. And I'm just going to now, I don't know, again, use my hot soldering iron here. Okay. And let's see if some of them actually are able to melt and maybe others not. Yeah, see, for example, those, they don't quite melt as much. So this could actually be, some of them, yeah. I've actually have found a few of them just today in the afternoon, which then actually did melt and then actually smeared, on the microscope slide. Yeah, so sometimes. Okay. So this one, for example, yeah, this long one, for example, does not seem to respond so much. Yeah, they seem to be these here. Most of them, don't seem to melt, but some of them in here might. Yeah, and then they kind of pull together, and then everything sticks together. This is actually a more I think a method problem that I'm having here right now. It should be a little bit more separated, I guess, so that I'm able to pick them out individually. Okay, let's try this here again. This one over here. Yeah. No, see? No, does not seem to respond so much. Yeah. So what I think is indeed up. Oh, okay. Now it got stuck. Let me try over here something. Yeah, they're still very much together. See this one over here. This seems to be on the other side of the slide. Yeah. Okay, I think most of them over here, I think most of them over here do indeed appear to be non-synthetic. Yeah. But let's have a look at some over here maybe. And so you can kind of play around. And what I would like to do a little bit is I want to optimize this a little bit and make sure that the tip is a little bit. So maybe I'm going to file down the tip a little bit. And because this is actually also a nice experiment, to do a little bit of citizen science. How much of the dust actually is basically made of of plastic, so to say, of microplastic. Yeah. So, but I think I'll leave it at here right now. I just check the time a little bit. I do have over here this. I showed you this, and I do have just a second over here. Paracord cotton dust. Yes, another thing that I want to show you. Because a couple of years ago, I got, it was already maybe 3-4 years ago, I got an email and I said, oh my gosh, people are worried about really certain things. And I just want to show you, because they found those fibers and there was this, unfortunately, this misinformation online that those fibers are actually worms. I'm not kidding, okay? There was this, they called them Morgellons. I have no idea who came up with that term. These are basically kind of, these fibers are worms because they're moving around. And then basically I was able to demonstrate the following. You take a piece of tape, okay? You take a piece of tape and then what you do is you just, I'm gonna put it on my pants now, on my jeans, right? And I'm going to simply, you cannot see that, but I'm just collecting a little bit of whatever is on my jeans and I tape it down over here, right? Okay, and what we'll be looking now is at the edge here of the tape, yeah. And I just want to show this to you because this was actually a little demonstration because this is actually something that people were really worried. Let me put that soldering iron away. Otherwise, I'm going to burn myself. I just want to show this to you. Scope. I'm going to just use regular. Let's go do it like this, and it's exactly here we are. Morgellons disease is paranoia. It's a mental problem where people think there are parasites when there aren't. Yeah. And I basically showed them the following, that the fibers that they found are essentially just fibers. And I just did in this one video. This video must be still online a couple of years ago. And then basically I said, look, these are just fibers that are collected. So what we've got over here, that's the tape. And over here, that is the how do you say, the fiber sticking out. And what happens is when you breathe on them, sometimes actually because they absorb moisture, they start moving, right? This is something that they have seen, and then all of a sudden they thought, oh my gosh, these are moving worms. No, they're not. Yeah, those fibers just absorb moisture, and therefore they start deforming. I don't know if I'm able. Yeah, look at this. Did you just see that? Yeah, how it's moving? I was just kind of breathing on it, and then as it dries, it starts to move again. Yeah. Okay. You can make those fibers move, and this actually freaks out some people. And I'm saying, come on, man. Yeah, sometimes it's just dust. Look how cool they're moving, right? And they're moving even after I've blown on them. And the reason is because my moisture kind of, they absorb that. It's also kind of a sign that these are kind of natural fibers. My jeans, that's made of cotton, right? So they're kind of absorbing the moisture, yeah? And that's it. There's nothing more to that, yeah? Yeah, here again. Yeah? So let's try it again. Yeah, then you have them move around a little bit. Yeah, as they absorb the moisture. And then when they dry, they kind of sometimes move back. Yeah. Yeah. I'm reading the comments here. There was a whole paper on it by a quack scientist who used raw helical algae claiming nanobots. Oh my gosh. Okay. Now I tell you why I'm kind of worried about people worrying about these things, because they're actually real things to worry about, in real environmental pollutions and so on. Yeah. So I always say, please worry about the right things and not just about fantasy things. Let's make the worms move again. Worms under quotation marks. Yeah. See. So again, they're not. I'm not blowing at them anymore, but they keep on moving afterwards, right? Because of the, as I said, as the moisture starts to evaporate. And I mean, I've shown. Basically, I answered then the email a couple of years ago, and then I actually also showed how real worms look like. I mean, real worms look like real worms, right? And not just like these things here, right? Yeah, just saying, yeah. But that's all, yeah. Let's add a little color because everything's so nice. Yeah. So another thing that I just also wanted to show you again, in case. Because I've not tried this as well. So again, this is that's basically the stuff from my genes on. Yeah. Then when you put it again across here, then you have a nice combination of color from the tape, and also of course of the fibers. And here you can see, again the edge of the tape. I'm reading now some of the comments again. Yeah. So yeah, here we go again. Yeah, it's sometimes it's kind of fun just playing around like that. Yeah. So let me have another look at a few things that I just want to show you, because we've done a lot of fibers now. 46 minutes. Yeah, so what I want to do now, maybe, I don't know, maybe I'm just going to move along a little bit and show you some biological samples, maybe a little, how do you say, change in topic if you want to. I've been through all of the fibers I actually wanted to I mean, I've got some house dust from 20 years ago, which really looks pretty much the same. No, I will show you something. Just as a little bit of a, how do you call this? I just want to show you how my little pet is doing. I know that's going to be now a little bit of a, how do you say, change in topic. Where is it? Here it is. You remember this little guy? Okay. Still alive. Okay, just wanted to keep you updated. Yeah. Spiky. Yeah, I don't know. This guy has been going on around now since October, and every time when I put it under the microscope, it doesn't seem to like light. Okay. If you've not seen it yet, I'm going to show you something else. First of all, interesting. Do you actually see those tiny little hair, right? As well. But I want to show you again a little bit just look at the back here of this little animal. I'm going to carefully touch it with a toothpick. Let me see if I can do that here. Let's see if it still responds. Did you just see that? Yeah, that's a defense mechanism. Okay, which I showed it to you already some time ago. Yeah, and no, I don't give him water. Interestingly, and it seems to be doing fine. Many of those animals, I mean, I was feeding it a little bit of my fingernail because it likes to eat protein, but many of these animals are actually able to produce their own water when they are respiring. It's called metabolic water. So when they do cell respiration, then they will actually also produce their own water. And I just want to, you never saw that part move like this. I want to talk a little bit about that because this actually relates a little bit to the fiber part. Okay, so you've just seen the back part here. Here, I turn on the arrow. The back part here fan out. And something that you don't see at this magnification very well is that actually it looks like tiny little arrows. So I'm going to zoom in. Okay. And even here, you cannot quite see it as nicely. Yeah. But those tiny little hair here, at the tips, they are a little bit larger. Okay. Over here, you can actually see that it's almost like an arrowhead that they have. So what I've done, I've actually found, I don't know if it's toxic or not. Okay, this is a so-called a carpet beetle. So it's the larva stage, and apparently it's not good if you find them in your home because they eat carpets, because carpets are made of wool, which is an animal fur. I put a lid on top now, okay? Just in case you're wondering. So, and what I've done now is I'm going to show you now some of those here, because I've also been able to find them in some dust samples as interesting fibers where I did not know what they were. I need to kind of I need to find them now. Just a second. You always start at a low magnification, maybe. Here they are. Okay, so again, they look a little bit like fibers as well, but these are those tiny here. Okay, so-called setae. And when I found them first several years ago in a dust sample, I had absolutely no idea what in the world this was. Yeah, I had absolutely no idea. Yeah, until I posted a picture in a German Microscopy forum, and then somebody actually told me what they are, and that's basically how I was able to identify it. Okay. Nanomachines, exactly. Here we go. So if you want to be afraid of something, how is that? That's actually a good one. I mean, they look like, I don't know, like weird, right? I don't even, are they not classed as a form of a fiber at all? I don't even know. They are called, I don't know if you should call them fibers. They're officially called setae. Setae are basically, how do you say, the hair of insects and yeah. Yeah, so, and in any case, I found actually quite a few of those, and even in a commercial microscope slide as a contaminant. Yeah, so yeah, I know it's written with an S, the setae, and that's a general term for hair that you find in arthropods, insects, and spiders are called setae. Yeah. The thing I have to tell you, what fascinates me a little bit is how in the world do they even grow? I mean, this is the thing that kind of, they appear to be non-cellular, right? Yeah. So that's the question. I don't know how, if the top part kind of grows and as it becomes longer, I really don't know. Yeah. Yeah. For those of you who speak German, those here have a very nice name. They're called in German, they're called Pfeilhaar. Pfeilhaar means arrow hair, if you translate it into Pfeilhaare. In German language, sometimes you actually have very descriptive. You have very descriptive nouns. Yeah. Okay. So this, honestly, this is something I'd like to explore a little bit more. I don't know if I have the book here. Just a second. Just give me 10 seconds, please. Yeah, I do have the book here. I mean, I just want to show this to you. Honestly, this goes back to the beginnings of microscopy. When I don't know, a long time ago. I'm just going to show this to you because this is how the book looks like. I actually made pictures. This was when did I draw this? Look, 1998. This was quite some time ago. House dust. Oh, okay. Oh, that's nice. 5798. 57-2. Oh, look, I've got the slide here. Look, 5798, 5798-2. These are basically the coordinates. And this is basically what I found here. So basically already back in 98, I found this weird thing here. And I kind of made a drawing. Unidentified, I wrote down here. Okay. Now, of course, I know what it is. Yeah. So let's have a look at this slide from 98. I don't know if I'm still able to find it. Yeah, because so this is one of those house dust slides from 98. Gee, oh, this was over 20 years ago. Let me quickly have a look. Where is the scope? Microscope. So that's the house dust again. Just a second. Let me see. I don't quite remember exactly where it was. I found it. I found it. Are you able to see it? Yeah. So this is a nice permanent slide that I made. So it's here. Here it is. This is now in DIC again. Yeah, let's just go higher yet. You know what? I'm gonna take this out here. Here we go. Here it is. Yeah. So that was actually the first time I found this in the dust, right? And then I had absolutely no idea what in the world it is. That's basically the picture that I made here. Okay. You know what? Let's try phase contrast. Just for the fun of it. Okay. Yeah. Yeah. So. A chitin is not a protein. It's I think a polysaccharide. Okay. But I wouldn't be surprised if there are certain proteins. How do you say included in there as well? Yeah. Yeah. So I bumped it. So that's kind of just yeah. Let's go up yet higher. 60 times. So dust can be quite an interesting specimen here. Yeah, yeah. So yeah. So I made a couple of I then stopped drawing because it was kind of very time consuming. Yeah, as well. But I did make a couple of drawings, and yeah. This was just at the time. At that time we didn't. I don't even have a. I didn't even have a webcam. 5.7. So okay. There's another one. Another thing I found here. Just let me. I don't know if I'm able to find that one. Let me see if you're able to identify what it is. Let me go down again. Where's the magnet? Let's further down. And I don't know if I'm able to find it. That's again the fibers here. Here it is. Yeah, that's the thing. That's the thing here, right in the center. Yep. This one I found out fairly quickly what it is. They are also quite common. Those weird structures, and you probably want to see the comparison here. Here, that's the drawing. Yeah, back in 98 as well. Unidentified structure I wrote down here. It's not unidentified anymore. It's a trichome, yes. Yes, it's a plant trichome. The plant trichomes are plant hair. And they are used also as defensive structures. Sometimes they break off quite easily. And yeah, I have to say again the German term Sternhaar, basically, which means nothing but a star hair. Again, a very descriptive term. Yeah, so that's how it looks like in bright field. Yeah. So that's one of the nice things. Thank you very much again for the questions. I got some questions because there are now some off-topic questions that I also wanted to talk about here. Yeah. Also commonly pictured by those with more, yes. Yeah. So yeah. Honestly, because this is, I consider people who can draw something very. I am not talented, honestly, and I have to tell you the following. I did an exact copying. So basically, I almost measured out everything. So this was not an, how do you say, a creative drawing. What people have done and before is there were those devices where you could actually project the image down on a paper so that you could trace it out. Back in the day, from a microscope. Right? So that is one way of doing it. Or a second way of how you can make drawings under the microscope is by actually looking at the paper with one eye and looking through the microscope with the other eye. Yeah? And then you can also try to basically trace it on the paper. Okay? Yeah. So this is yeah, just yeah, just wanted to share this with you a little bit because we were looking at some interesting structures in dust. Okay. Let me quickly. So let me quickly have a look here. That's true. Yeah, I will quickly read some of the comments here. Okay. Okay, thank you, Jesse Ever Smaller. Did again a little bit of research. So from a quick Google, it seems the setae are made up of multiple fibers made up of chitin, usually arranged in a helical structure. Okay, so basically these individual fibers. Okay, so that's not a how do you say a chitin here because it's a plant thing. Yeah. Did you make a solid sample of it and describe the slide nicely? So what I've made is the following. Back in the day, in '98, I made a permanently mounted slide. That's the one that I've, this one over here. Yeah. And what I've used is the following. I used a numbering system, basically with a date, 5th of July, 1998. And then I always gave it -1, -2, -3. It's kind of like a date and a serial numbers, yeah. And when I made the drawing, I simply have written down, of course, also the number, yeah. And because it's a permanently mounted slide, the specimen is preserved here. So this was at the beginnings of microscopy. I basically was just practicing making slides, and I collected whatever dust samples and whatnot, stuff that was easy to make. At that time, the mounting medium that I used was known as a U-kit, which is not healthy because it contains an organic solvent, but it was a very fast drying one and a very stable one. So just, yeah, I wanted to share this with you as well. So I'm going to just quickly read here. I got a pre-loved Olympus stereo zoom, and I'm forever grateful for your advice video on new microscope versus refurbished best brand. Okay, thank you. Yeah. So let me quickly check again. Otherwise, can you test UV reactivity? That's an interesting one. Do I have my? I don't know. I mean, I do have a UV lamp, and this UV. I don't know. Honestly, thank you for asking. I'm just gonna try it. I guess. I'm just gonna use any old fibers. I'm gonna turn off the light completely. So, and I'm going to turn on the stereo view. So these are basically, yeah, you see the light that you see, while you're still able to see this, that's just the ambient light. Let's turn off the arrow here. Let's focus it. So this is kind of a mix of fibers. And I'm just going to shine on the UV light. This is, I don't know. It looks blue. Maybe you have to dye it with something. There's something that I might try out. Do you know those yellow highlighters? Yeah, they do shine, and sometimes it is possible to stain specimens with those. But you see those here, they don't seem to be very. This one doesn't seem to be very interesting here. What about this here? I don't know. Let me try those here. Now I'm also a little bit. Now I also got a little bit curious. Okay, I'm going to try those here now. Those synthetic fibers from the cord. From this one over here. If they are fluorescing, I don't know. I dropped it. Here it is. I need to switch over again to the stereo view. Again, it's not entirely dark in my room, but I don't know, maybe a little bit. You know, it could be just also the reflection. No, but real fluorescent light should be much brighter. Again, something that I also have not yet optimized yet is getting algae to glow, but because what they do is the chlorophyll also starts to fluoresce, and however, you need to do a long time exposure for that. Yeah. So what I'm gonna do now is I'm just going to, I don't know, maybe put in a nicer sample so that we have something to look at while I go through a couple of the questions. Because there are some off-topic questions as well that I want to talk about. See, this is way too bright. Okay, here we go. So that's again. So let's have a look here. How can you find out if some fibers are natural or synthetic? Yeah, very briefly, the synthetic, basically I mean plastics like polyester, nylon, and so on. The melting test is one thing, and there are also special stains that you can use. And very often synthetic fibers are not so irregular. So this one basically are very irregular, so they're mostly natural, I would say. Okay, so that is the problem with the DNS hosting provider, with my internet service provider, because it's some issues with my web page, or still have. Okay, so I'm going to skip over that. Okay, I don't have Kevlar. Kevlar. No, I don't. Well, Kevlar is that's an interesting one. The reason why I'm thinking is the following. This was over 30 years ago. Really, I was still at university. 30 years ago, I did paragliding, with this chute, and you're kind of running down the mountain, flying off. And the lines of the paraglider were made of Kevlar material, and these were extremely strong fibers. I wonder if I still have a couple of those lines somewhere. Yeah, but that would be interesting. But Kevlar were very fine fibers, but were extremely strong. Yeah. Okay, I don't have natural cotton, unfortunately, right now. This was another question. But it gets me wondering why washing machines have settings for synthetic cotton and other materials. Yes, I might be able to give you a little bit of an answer here. Is this because washing machines can be quite hot? And synthetic materials, as we've seen, are temperature sensitive. And this means that if you actually wash temperature sensitive materials too hot, then it might actually deform. It melts, so to say. Yeah. And that's the thing. In cotton, for example, that's also one of the reasons why underwear and so on is made of 100% cotton. You can essentially wash it very hot. You can theoretically even boil it, and it's going to be fine. Yeah. But if you try doing that with synthetic materials, then very often they start to melt in the washing machine or deform. Yeah. Though the asbestos question I talked already about. I said about marine aquariums on your last stream, I had a coral being irritated by something. I took a sample next to the base of the coral and it was a hydroid. One of the things that I've also done, and I have not found more of those concerning water samples, I'd like to again catch a couple of hydras. They look very nice also under the microscope. So maybe in springtime again when I will try to find some in my local pond. Okay. So the setae, and there was a question about a microscope camera, okay, or fiberglass, okay. I do also have. Would it be a cool idea to watch bacteria being killed by UV light under the microscope? First of all, yes, UV light does kill bacteria. I did some experiments already, and UV light is actually used because to disinfect rooms in hospitals and labs, for example, overnight to turn on UV, and this will actually kill bacteria. However, I don't know if you're able to see it that well, because some bacteria, it has to stop movement, and I doubt that this here is strong enough. Because this is just a UV flashlight, but you see, it's sufficiently safe right? But the actual really strong UV light, which is able to do cellular damage, you'd actually get a sunburn from that, right? And actually in the lab we did use those as well, but we had to wear protective masks and gear not to get any skin exposure, because they told us, they warned us, if you're getting any skin exposed to the UV light, because we used it for DNA staining. Yeah? They said that within 30 seconds you're going to get the worst sunburn you can ever imagine. Yeah? And this is this UV light over here is actually very weak. Yeah? So I don't know if this one would be enough to actually have an effect on bacteria. Yeah? It might be too weak. Yeah? There was a question about a microscope camera. Did we note what mounting medium? On those slides, right? No, I did not write it down. See, at that time I didn't write it down. I thought I didn't even know that there were different types of mounting media available. Why did it switch this off now? Here we go. There was a question about a microscope camera, and that I said I want, and I tell you the following. This is a complicated question, right? Basically, what I would suggest is there is no ideal microscope camera. And a couple of months ago, I did make some videos, some live streams even, so you might want to go back a little bit and check this channel, where I presented a couple of adapters, also camera adapters and so on, to give you an overview. It is indeed like this, that you do not need absolutely a microscope camera, such a dedicated one. You can also work with mobile phones. However, it is by far not as convenient. Okay, there's one thing that I need to clarify a little bit. I'm showing all of those nice colorful images and so on, and people think that it's the camera, right? It's not, right? It's just that the sample is well prepared and it's flat. And then sometimes I also use, very often, almost always, I also use Photoshop to enhance the contrast. So essentially, whether something looks nice under the microscope or not is not a camera question primarily, right? It's a specimen, a sample question, and also a contrast question. Yeah. So a UV tube lamp is cheap enough, yeah, but I know, and actually we have one also in school as well. But the thing that I just want to say is, in that case, I would need some protective gear as well. Yeah, some face shield, UV shield, so otherwise I don't want to hurt my eyes. Yeah. Or there's an eyepiece camera. It's not amazing, but there is a, yeah. And indeed, there are certain eyepiece cameras which you can insert into the microscope instead of an eyepiece. So you do not need a microscope with a photo tube, even though that's way more convenient. It is possible to get those eyepiece cameras and then connect them directly over USB. However, you have to be aware that you will not always get a very smooth image at high resolution. The reason being that the USB connection sometimes is too slow. Yeah. So that's just saying, there is nothing is perfect. I've been using microscope cameras also for quite a long time. Yeah. Let me quickly have another check. Yeah. So you know what I'm going to do now? Because I decided to always occasionally show some living things as well. Some people want to see some living things. So what I'm going to do today for next week, in preparation for next week, is I need a small jar, which I've got over here. So I'm going to make another topic change for the last couple of minutes. And what I'm going to do now is the following. I'm going to do a little, show you how I make an enrichment culture. So, yeah. Thank you very much for the comments. So I'm going to show you something very briefly, and then I'm going to show you how to make a so-called enrichment culture, because that's also quite something that I'd like to try again. So I'm going to grab again my old little water sample. Here we go. This disgusting looking jar with this weird stuff floating here on top. Some of you might actually remember from last week. So let's put this under the microscope again. So I'm going to take something right from the very top. Okay, you have not seen that because it's over here. Cover glass goes on top, obviously. Cover glass goes on top. And under the microscope. Let's put this away here. Where is the scope view? Again, yeah. You might already see a little bit of some stuff moving around, some tiny things. I'm now using the lowest magnification, of course. Yeah, there's some movement going on. It's pretty active actually, but I need to go up with the magnification here. Larger paramecia I've not seen yet, but I'm quite sure that they're there as well. Whizzing around. Probably some of them might even be bacteria. No, for bacteria they're too large. So so we see at first check we do see that there is some movement on here, and what I would like to do is I would like to increase the concentration of those guys here. Especially also hoping to get some paramecia going. Here's some more, not paramecia, but yeah, other ciliates. Okay, you get the idea. Okay, so there is stuff present, but not, in my view, not in sufficiently high concentration. So what I therefore sometimes do is the following. I'm just going to show this to you, okay? I'm just reading some of the questions. I will go back to those questions later. But you need a jar, a small one, okay? And I now need to somehow transfer a little bit of the water over here. I don't want to pour it over. It's kind of disgusting as well because, yeah, I'm just going to use some, I don't know, pipette, I guess. Yeah, maybe that'll do. Okay, so just taking some of this stuff here. Just generously transfer. And again, I have to say, we're dealing with living systems here, and they sometimes are difficult to control. So sometimes it's a question of trial and error. A little bit some green stuff as well. Okay, maybe one more. So there'll be plenty of microorganisms in here. So this is kind of disgusting now. What I'm going to do with that, I need to rinse it or throw it away. I'm just going to put it somewhere because I don't want this to make any mess. So I'll put the jar away. And then I need some more tissue paper to wipe up the mess here. So, and then, I'm going to wipe this here as well. You see, because there are bacteria and microbes present and so on, it's not that I'm super scared of them. I mean, these are water microbes. I still want to adhere to basic hygienics. So, and then now here comes the important part. What you need is you need 2 spoons. We can make it a little bit larger. Okay. And you need this here. These are wheat grains, cereal, basically whole wheat grains. And you just basically need 1 or 2 of those. And basically this works far better than, for example, other cereals, because sometimes I just used regular oats, which is pretty much the same, but this works better. So because for stability, because they're kind of more solid. And then what you do is the following: you crush it. Oh, okay. And you drop it in here, and then you wait 3 days. Okay. You may want to push it down, and what happens is the following. Okay, this contains a lot of starch. Bacteria are going to start growing there, and then the ciliates, the paramecia, and the larger ciliates, they will feed on the bacteria. So you have a small food chain, and then after about 3 days, 3 to 4 days, you have to wait. You pick out the. You're gonna see that it's going to start to become very slimy. And then what's going to happen is there in the slime layer, which seems to be maybe some kind of a polysaccharide, you find a lot of these microorganisms, especially paramecia. Yeah, I tried it with a rice grain, and it did not work quite as well. And the reason, I don't know why, but I think, again, I'm guessing, is this because in the how do you call this, in the seed coat, in the brown seed coat, there might be some nutrients present as well, I guess, or it's like this, that on a rice grain it tends to fall apart, right? But the seed coat kind of keeps everything a little bit together. I just want to show you this one. You also know from a previous one. Here I did not use the whole grains, but I used the following. Just a second. I dropped in just regular cereals, yeah, the breakfast cereals, right? And they did not. I mean, it also works, but they have the disadvantage that they start to fall apart. Okay, and then what happens is that there is a little bit of an overfeeding happening, and then the whole liquid starts to it breaks apart, and then there are bacteria all over the place, and not just concentrated around the cereal. And those white things that you see here, these are those. And what it's now a little bit more difficult to actually take out a sample from there because I cannot go in with a tweezer and pick it out and dip it on my microscope slide because it falls apart. Yeah. For convenience and for I found this with a crushed cereal grain to be way more effective. Yeah. Even though, yeah, this one should also work. And then, yeah, what we can try now as well in the last couple of minutes, still have a look if any changes happened from last week. So again, I need and then I'm running out of microscope slides already. Have to kind of wash them again. So what I'm doing then now is the following. You go in here. You kind of go directly into the cereal. Yeah. And you take a small sample. It's a very small sample. I have to discard the tip here because tips that were in the in this liquid, in contact with bacteria, I don't want to have floating around. And the cover glass goes, of course, on top. And then now we can hope that there is still plenty of them present, because if there are too many bacteria in the sample, then the oxygen concentration will rapidly drop, and then all the larger microbes like paramecia and so on are not able to grow. And we don't want that. Okay, let's have a look if there is more going on here. Yeah, oh, this one is dividing. Look at this, isn't this cute? We were lucky. Yeah, that's a dividing one. And I have to tell you, in a cereal grain, I have seen concentrations that were way, way, way higher. Okay, because somehow in this slime layer that forms, it's just going to be full. But this is actually something which shows that there's still division going on. Yeah. Okay. Yeah, it looks quite nice. Let's have a look. So honestly, I've seen those already before dividing. You might have to wait another hour. Yeah. So. So the question is if those are lacrimaria. I mean, they also have very long, yeah. Maybe they're related. I don't know. And I don't like the color. I think that's better. Oh, and in the background, tons of bacteria. Those tiny things that are moving around. Yeah. Yeah, and those lacrimae, they have very long necks. Sometimes I've seen it. Sometimes they've also retracted them. They can retract them, but yeah, lacrimae look very, very distinct. This is actually quite a nice one, I think. But unfortunately, this is still gonna take. I mean, we can follow it along, but I think it might still take some time until it divides. But you can actually see it becomes a little bit more narrow already here. Maybe we're just gonna follow it around for another 5 minutes until the end of the live stream. Yeah. Yeah, and that's essentially the thing that I kind of want to have is I want to have a very high concentration of those, and then you can do a whole range of different experiments with them. For example, electrotaxis, you can apply a voltage across it, and then you're going to see that some ciliates move along the voltage. Yeah. Let's see if I'm able to. Let's do it like this. Ah, look at this. You see, some parts are actually polarizing there as well. You see those yellow dots in there. And in the background, all of those things that you see, those tiny things, are all bacteria. So, this bacterial soup is actually not ideal because they take away so much oxygen, right? With a crushed cereal grain, everything is a little bit more concentrated around the cereal, and then the liquid around it also looks much more clear and not quite as cloudy. Yeah. So that's why I want to see how well this actually works this time with the crushed cereal grain. Usually, again, you have to wait around 3 days or so. Yeah. Yeah. I'm just reading again a couple of comments here. Yeah. Yeah. Okay. I'm actually quite happy that. But now I actually want to wait until it divides. But then again, I've observed these things before, and it took a long time. But actually, you see that. Yeah, they're almost separated now. You can also sometimes what you can see is the following. This is now cell division, obviously, but sometimes they will also conjugate, and conjugate means that they are exchanging DNA, and then they will line up in parallel with each other, and then they're exchanging DNA. Yeah. What's your previous question? This is 60 times. Okay, I need to quickly go back to your previous question. If I can find it. Did you get my recent email? Ah, yes, thank you. Yes, I got your email. I did not respond to it, but I read it. Yeah, thank you very much about the enzymes in the sample. Okay. Well, the question was is whether the enzymes were present already from the beginning. The sample from last week's live stream about the spermatozoa. If you have not watched it yet, I can highly recommend it. I think, as I mentioned also last week, I think one of the nicest microscopic videos of spermatozoa that I've ever seen. Yeah. Yeah, yes, it made it through. Thank you. Sometimes, look, another one over here. Sometimes I get a lot of emails, and then I have not always responded, so I'm already behind with the response over a week. Yeah. So actually, I do want to follow this along now. But you see, there it's almost divided. It was much. They were much more connected before, so it starts to become thinner and thinner and thinner, and then they separate. And one of the nice things here is well that you can see the cilia moving. Yeah. Yep. I don't know what I'm gonna do now. We can keep on watching this for I don't know how long. Yeah, I remember a couple of years ago, I watched this for, I don't know, 1.5 hours. Yeah, and then finally they kind of separated. Yeah. But it would be a pity to switch off now and say goodbye and then, yeah, see you next week. Crystals, yeah, I think the question is why are these crystals? Because you look at this. Yeah. There must be something in there that's kind of polarizing, and I know that people have already used them under cross-polarized filters and they've seen things. I don't know what they are. Yeah. Look. Oh, very nice. I mean, look, 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 look. Look, 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 look. 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 Ha! They separated. Or are they still a little bit connected? No, I don't think that these are microplastics. Okay. Here we go. Ta-da! Yeah. Here we go. Let's go down a little bit with the... Okay. Yeah, what I have to say is the following. Why, and I know I'm going to close off with a little bit of philosophy again. The thing is what fascinates me is, of course, now the concept of individuality. You know, you've got one organism now, right? And then you've got two genetically identical organisms. Yeah. 2 times the same. Which one's the parent? Which one's the original? Which one, is the offspring? It doesn't apply anymore. Yeah, at this level, it's completely different. Yeah, so all of those human concepts, yeah, that we have of individuality, yeah, simply don't apply. Yeah. Okay, didn't expect this. Now we've also seen a cell division. We've also seen a cell division now as well, right? So, and that's essentially one of the things. Is if you want to also observe these things, I highly recommend those enrichment cultures, because then, of course, they reproduce in there. But I just also want to mention, please observe hygienic standards, because as you can see in the background, plenty of bacteria. Maybe that's the last thing I want to show you, simply to show you that there are, yeah, all of those dark dots that you see floating around here in phase contrast. These are bacteria. So a lot of them, right? That's why you want to be a little bit, generally careful. Spiral-shaped bacteria, even, yeah, spirochetes. So you want to observe general hygienics here. So what I want to do is wear gloves when I prepare my slides for moths. I think maybe that's a little bit not necessary. We're exposed to when we walk on grass or whatever, you know, we're always exposed to microorganisms. Yeah. In any case, I think I'm going to call it quits for today. We started with fibers and we ended with a cell division. Also a little bit unexpected. Yeah, hope it was interesting for you. Please, feel free before the start of the stream, if you have any specific requests of what you want me to put on the microscope, please just mention it. No guarantee that I have all of the samples prepared and ready to go, but maybe sometimes I do. We'll see. Yeah, I wish you all the best. Okay, hope to see you again next week. Happy microbe hunting as always, and all the best. Bye-bye.